So antigens bind to antibodies via the induced fit model of protein ligand interactions. And if you look at these different models, we've got our we've got our protein conformation when no antigen is bound. When the antigen is bound, but the antigen is not present, one of the things that I'd like you to see is, and this is something that you as scientists, I'm sure, have instances where you talk to somebody in your family who's not a scientist or a friend or something like that, and you're like, oh yeah, it's so cool, and they're just kind of like, I miss the connection there. This, I think, is the sort of thing that structural biologists encounter, where they're saying, oh, this is radically different, this figure in model A versus model B. And there are subtle differences, but those subtle differences cause a huge impact. You know, hemoglobin is an example of a small change causing a huge difference. Binding to one O2 molecule causes ultimately four O2 molecules to bind. So what we've got is, and you can kind of pinpoint this versus this. Well, the point that I've labeled A has swung out. B hasn't really changed too much. Maybe it retracted inward, and the entire kind of back of the protein looks a little bit more electronically dense. So we see these changes that have taken place to our protein, and here is the antigen that's actually bound to our antibody. Now, the thing that we're not shown here is what the antigen looked like prior to binding, but what you can take away from this is that that antigen did cause some change in the overall protein structure. So there is that induction that the antigen brought about to the protein. Now, an antibody specificity is an important analytical reagent. So the way that you can do this is known as an ELISA assay. And what you can do is you can use a surface or a plastic surface or something like that and coat it with sample antigens. Here we've got this, and I realize now my head is blocking the orange one, but you've got these unique antigens. I'm going to label them A, B, and C. I know that I don't really need to do that because they have color indicators, but what these individual antigens are going to be applied to the surface. And what they're going to be able to do is they are going to adhere to that surface. And what you're going to be able to do with this is known as an ELISA, as I said earlier, ELISA, enzyme-linked immunosorbent assay. You're going to be able to tell whether or not you have a specific molecule here, a specific molecule present. So what you will do is you will coat a surface with specific antigens. Then you're going to wash that with what's known as a blocking solution. Then you're going to incubate that surface with a primary antibody. That primary antibody will detect, in this case, we're detecting antigen B, and it will adhere to that. When that is present, then our secondary antibody, which has an enzyme attached to it, will then bind to our primary antibody. So what we're doing is we are probing for the presence of molecule B, and we are probing to determine whether or not our antibody is functional. So if this antigen B was present, or so long as antigen B is present, the primary antibody will bind to antigen B. And if that primary antibody is present, our secondary antibody will bind to that primary antibody, and that secondary antibody has an enzyme that is covalently linked to it. You can add a substrate to that, and then you can produce a product which gives you the presence of, it's kind of an indirect measure of whether or not that initial antigen B was present. Here's an example of what an ELISA assay looks like. You can make these quantitative or qualitative. If there's any subtlety of a yellow pigment there, you can say, oh, well, absolutely, there is indeed some of my antigen present, because the only way that I got an assay, or the only way that I got a positive result, was if my antigen was present, because my antigen is going to bind to my primary antibody is going to bind to my secondary antibody. And that's what an ELISA assay is. So our third case study for looking at protein function is to look at muscle proteins. So, and the structure of those muscles. So muscle fiber is a large, single, elongated, multinuclear cell. Each fiber contains approximately 1,000 myofibrils, and what those myofibrils are, here is an individual myofibril, and I realize my head is blocking the actual proteins within there, but it's kind of these long chains and these different colored chains. We're going to look at those in just a second. The things that I'm blocking are these I band, and there's something called a Z disc and an M line. I'm going to show you that on the next few slides. So myofibrils contain thick filaments of the protein myosin. Now, myosin looks a little bit like this in that it's multiple proteins that have two supercoiled alpha helices. And these two supercoiled alpha helices have these N-terminal heads that then have these light chains attached to them, these light chain proteins attached to them. So myosin is so you have these repetitive myosin proteins supercoil around one another. Now, myofibrils contain thin filaments of the protein actin, and these thin filaments of actin kind of stretch along the length of those myofibrils. Myosin thick filaments slide along actin thin filaments. So here's a myosin head group where those two different heads, basically the N-termini of our heads, are going to attach themselves to the thin actin filaments. And what this is also going to, or what this is ultimately going to do, if I could draw these in a little bit. So the color, let's see, this is our myosin, and these are actin. Okay, so all of those myosin head groups, they had almost little hooks. Those hooks that could basically latch onto and subsequently release from the other protein, actin. So myosin has those head groups that interact with the actin filaments. What they can do is kind of cause a contraction and a relaxation. When they're contracted, that means there's overlapping of those actin filaments and myosin filaments almost 100%. Whereas whenever they're relaxed, well then the myosin is basically overlapping only a fraction of the actin filament. Now, protein interactions that are modulated by chemical energy, actin and myosin interactions are indeed modulated by chemical energy. So the use of ATP can cause conformational changes in proteins, which are generally required for their function. So motor proteins are prime examples of this. So muscle contraction occurs through a series of conformational changes to protein structure due to the binding hydrolysis and release of ATP and subsequently ADP. Because if you remember, ADP is hydrolyzed into ADP. So in order for this to happen, we have this Actomyosin cycle that explains it all. So we've got a myosin head group that has bound to one portion of a actin filament. That myosin head group is going to be a part of actin, which is going to bind, that myosin head group is going to bind to ATP, which is basically going to interrupt the interaction between myosin and actin. So here, my left hand here is my actin filament. My right hand, or my right fist, is my myosin head group. Now, in order for this to bind ATP, it's got to release. It's going to then bind to my ATP, and it's going to hydrolyze ATP. It's going to cause a change in the structure, And since it's released and it's hydrolyzed ATP and caused this change in the structure, it's going to move on. Okay, so that myosin attaches to an actin filament downstream, which causes that PI release. So the hydrolysis of ATP has taken place, but now this myosin head group is bound to ADP and PI. When this is released, then it can rebind, or sorry, when it's released and it can bind and then release ADP, now this myosin head group is going to be able to bind to another part of our actin filament. So we've got them kind of color-coded here as a sequence. So we've got this darker orange and then a lighter yellowish orange, our initial site to our second site. And it's all driven by ATP hydrolysis and this change in the structure of the protein that enables kind of this movement downward on this actin filament. So the availability of myosin binding sites on actin is going to be regulated by a couple of different proteins. Troponin and tropomyosin are going to avoid continuous muscle contraction, and a nerve impulse is going to trigger the release of calcium ions. This release of calcium ions causes conformational changes to the tropomyosin troponin complex, exposing myosin binding sites. So whenever that release takes place, those myosin binding sites are going to be revealed, in which case myosin can subsequently bind onto that. And what we've got is kind of a display of what we're looking at. We've got troponin T is this sort of yellow or orange yellow or orange filament, and this orange section right there. Troponin C is this other portion of it. Troponin I is going to be a even different component, or a separate sphere altogether. And so whenever calcium is going to be released, well, this troponin tropomyosin interaction is going to be disrupted. And tropomyosin is this kind of filamentous protein that wraps around the entirety of the protein. So whenever that is disrupted, then myosin can rebind to the actin filaments. All right, well, I hope this is helpful. Chapter 5, it's a behemoth, but it's all there. All right, well, if you have any questions, please feel free to email me. And that was the email. So have a good one.